We will move to our next speaker, who is Javier Orozco Cordoba, who's coming from the Institute of Cellular Physiology in Mexico. Javier is going to talk about how TRIM3 regulates Th9 differentiation and in vivo function. So I hand it off to you, Javier.
Yep. Thank you. Okay, so good morning. Thank you very much for the opportunity to present here in this Congress. So today I will be presenting the work I developed during my undergrad, what I was doing working in the Licona Limon lab at the National Autonomous University of Mexico, and it is titled as 333 regulates Th9 differentiation and in vivo function. So as way of introduction, we all know that naive CD4 T cells can acquire different phenotypes that rely on the stimuli that they are exposed to. Canonically, we know that we have Th1 cells, Th2, T17, Tregs. However, in this project, and I will be introducing the Th9 cells that, as the name indicates, they do produce the cytokine IL-9. So Th9 cells and IL-9 may have different functions that rely on the context that you study them. However, in this project, we are particularly interested in the ones that have a protective role. So, for example, it was demonstrated that Th9 cells are key drivers of the anti-helminth response. So, through secreting IL-9, they can promote the function of different innate cells, which then will promote this anti-helminth response. On the other hand, we don't know that Th9 cells can also secrete IL-9 in tumor contexts, so they can enhance the function and expansion of these innate cells, and also the function of CD8 T cells, in thus contributing the anti-tumor response. So to differentiate these Th9 lymphocytes, what we need is, as every other T cell, we need TCR and co-stimulation, but we also need the presence of two key cytokines, such as TGF-beta and IL-4. So if we only have TGF-beta, we will have Tregs, and if we only have IL-4, we will develop Th2 lymphocytes. So when we have both cytokines, we will eventually differentiate these T cell lymphocytes. And in the lab, we were particularly interested in study the TGF-beta pathway that starts with when TGF-beta binds to the receptor 2 and then the receptor 1, who then downstream the signals through SMAD2 and 3, which is phosphorylated and then can interact with the protein SMAD4 and be translocated to the nucleus and regulate certain type of genes. This is a canonical way that we all know and we study. However, there are other non-canonical pathways that can also exist. However, we are interested in the one that SMAD2/3, instead of interacting with SMAD4, can interact with TF1 gamma or TRIM33 in this context, and then regulate other set of genes. So this is the pathway that we're interested and we evaluate it. So TRIM33 is a multi-domain protein, so it has a domain for ubiquitin ligase, B boxes for protein interactions. In the linker, it has a domain for SMAD binding, and in the C terminal, it has two epigenetic readers, such as PHG and bromo, that can recognize certain epigenetic markers in the histones. So the functions of this protein is just goes beyond just interacting with the SMADs, so they can perform other functions. So this protein has been mainly studied in non-immune cells, and this is where it has been described. However, in the last years, we have been published several papers regarding the function of TRIM33 in immune cells. For example, we now understand that in dendritic cells, TRIM33 regulates the differentiation and stability of these cells, while in macrophages regulates the transcription of interferon beta during activation. Also in macrophages and neutrophils, it is important for the recruitment of these cells into the inflammatory cues. In B cells, it's important for preventing the apoptosis, and specifically within CD4 T cells, we do know that TRIM regulates the pro-inflammatory and stability of Th17 cells, and as Paula already explained during her talk, it regulates the stability of Tregs during inflammation. So we don't know what's happening with the rest of cells, but in this project we focus on the Th9 cells. So given the importance of TGF-beta on the generation of these cells, and that is unknown, this non-canonical pathway, we wonder what was the role of TRIM33 in these Th9 cells. So to study this, what we did is that we have a conditional knockout mice that is flux for TRIM33, and it is depleted by the expression of CRE, which is in the promoter of CD4. So with this we do deplete TRIM33 in all the T cells in the mice, And at the same time, this model is a triple reported. So we have GFP every time IL-9 is expressed, human CD2 for IL-2, and RFP for FOXP3. Um, so this is really convenient because we don't have to do any intracellular staining. So we can check, we can see the Th9 cells in this way. With the results, what we started is that we sorted naive cells and then we differentiated them in presence of TGF-beta L4 as well as other cytokines important for the expansion of these T cells. And 5 days after differentiation, we do analyze them by flow cytometry. What we found is that the DH9 cells that are deficient in TRIM33 have a better differentiation both in percentage and in numbers, and they do uh, produce more IL-9 measured by the MFIL of GFP. So with this, we concluded that TRIM33 is inhibiting somehow the Th9 differentiation and IL-9 production, and with other experiments, we also proved that limits the acquisition of this phenotype throughout the time. So in order to understand why we have more differentiation and more numbers of these cells, we do evaluate the proliferation as the main responsible of this phenotype, specifically on the Th9 population, and indeed we found a higher percentage, a higher index related to proliferation in these cells. So given that we have more of these T cells and they do proliferate and they secrete more of the protein of the cytokine that is important in the immune response, we wonder what was the in vivo function of this. So we use helminth infection to model this. So we inject Nipostrangulus brasiliensis helminth subcutaneously, and we do analyze 7 days post-infection in the small intestine, as well as doing flow in different organs. What we found is that when we challenge the knockout mice, they do have a smaller wart burden, suggesting that they have a better anti-helminth response against this ailment. So TRIM33 somehow inhibiting this. We do flow cytometry. We found higher numbers of Th9 cells in mesenteric lymph nodes as well as in lung, which is also an important organ and active inflammatory cue in this model. As I told you, IL-9 can promote the function and expansion of innate cells, so we do evaluate them. And we found that an increasement of ILCs in mesenteric lymph nodes. However, at that time, we didn't know if the Th9 cells were responsible for this phenotype because it was a CD4 model. So we did move to a clean model. So we differentiated the Th9 cells and we do sorted them and transferred into the right recipients, and then we challenged them with this helminth. And as expected, the mice that receive Th9 cells have an improved anti-helminth response. However, those mice that receive knockout cells may have an improved anti-helminth response. And when we do analyze these innate cells in these mice, we indeed found we found a higher number of ILCs and mast cells, recapitulating what we have in the whole mice. So we think with this that TRIM33 is limiting the effector function of Th9 cells probably in an IL-dependent way, given that these cells do respond upon IL-9. As I told you before, Th9 cells are also important in the tumor response, so we use a melanoma model to see what was happening in these mice. What we found is that they do have a better tumor response while seeing small volume throughout the time and smaller tumors at the final day of the experiment. And when we do flow cytometry, we indeed found that they have more Th9 cells in the tumor as well as in the tumor-dendritic lymph nodes. However, we need still to do transfer models in order to understand the impacts of TRIM33 in this population and what is happening with the tumor. So finally, we wanted to understand what was happening within the cell and why, what was TRIM33 regulating within. So what we did is we eliminated TGF-beta in the media of differentiation, this in vitro. And of course, TGF-beta is important for the generation of these T cells, so basically eliminate differentiation. However, those TRIM33 knockout cells do have more differentiation still, and they still do produce more online. So we think that there might be other pathways related or that have been regulated by 2033. So we did a qPCR to see if there were any transcription factors modifying these cells. And what we found is that they have more IL-4 and STAT6, both transcription factors associated to IL-4. So when we test this cytokine in culture, we found that when do not add IL-4 to the media, there's no difference between the wild type and the knockouts. While we only add 1 nanogram per ml, basically the TRIM33 knockout cells are more sensitive to this cytokine. And the same happens with the MFI of L9. We think that this is probably happening through IR4 induction. However, we still need to do some experiments to understand the mechanisms behind this. So with this, I conclude. We think that, of course, STRIM33 regulating the GFR pathway, but we also think that it's regulating the IL-4 by inhibiting STAT6 and IL-4. And this outcomes in a better anti-helminth response and probably anti-tumoral response. So we still need to do some other experiments and omics to understand the outcomes of this protein and to see if there are other pathways related to this protein. So I want to thank my lab. I want to thank Paula and Eugenio for their support throughout these years, and also any other member that, yeah, created a great lab environment. Thank you, and I'm happy to take any questions.
Amazing and fantastic talk. Congratulations. I guess before all this, what comes next with all of this data that you have together? What's your plan with all of this going forward?
Sorry, I didn't get.
Do you have any like next steps in this, or are you going forward with this data, putting it all together now, or what are you
No,
doing with
no,
the rest of the story?
I moved to another lab, so yeah, I'm currently looking
Okay.
for PhD. So yeah,
Oh, best of
hopefully
luck. That's
someone else will
exciting.
continue. One of my
There you
friends.
go. There you
Yeah,
go.
yeah.
Great. Well, we have a question here from Eleanor who is asking about the increase in TH9 ICs and MCs, if this increase by thousands is substantially different or meaningful within the data.
Yeah, we found a higher numbers. Probably you guys didn't see it a lot because the axis is in log, but yeah, we do see a lot of ILCs more and mast cells, especially in the rack model because they do not have any other signal. So yeah.
Okay, interesting. And I'm just about the IRF4 within the last slide you showed. Why do you think that IRF4 is that kind of connecting piece within that?
Yeah, well, what we found is that when we do differentiating throughout the time, we found that IR4 is expressed more rapidly than the wild type cells. And we do take these IL-4 curves where we modify the concentration. IL-4 is modified by the expression. It's IL-4 is modified by how much IL-4 we are adding to it.
Okay, amazing. And one more question here from Chaim as well. Have you seen the prevalence of these cells change after antibiotic regimens?
Yeah, no,
Um,
we
do,
haven't. Sorry.
no, just if they change at like a similar rate, if the kinetics of them are similar. If you have any comments on that.
Yeah, we haven't tested in non-germ mice. Yeah.
Sorry, in NSG or
No,
pathogen free?
we haven't tested in
Oh,
non-germ mice, so we haven't treated with antibiotics and in NSGs. Yeah, even though they cleaner, yeah, we do transfer them, so it's more artificial.
okay, amazing. Um, I don't see other questions coming in, but if there's anything in the chat, I will leave that to the audience to put out there. Thank you again, and congratulations, and best of luck with the PhD.
Thanks.
Now I will
Can I
pass
jump?
off to. Oh,
Can I just jump in for a quick question before?
Yep.
So I guess I didn't communicate too effectively. So when I was looking at your numbers on
Yep.
those cells, the increase in numbers, we were looking at like several thousand cells, and yet your commentary was that they had somewhat profound effects. I'm sort of scratching my head here saying, how can several thousand cells, when you're looking at those ILCs and MCs, how can what are your thoughts on that? Just a relatively small number can have that impact. Do you want to sort of speculate a little bit on what they're doing there that those numbers make a difference?
Yeah. We think that even though there are not a lot of cells and it's really hard to do a flow of these populations in vivo, we do think that they do produce a lot of IL-9, which is also very potent to promote the function and expansion of these T cells, of these cells, especially the ILCs that we do know that they expand upon IL-9 secretion. Hope that answers.
Amazing. Thank you again.